Are dragonfly larvae all about that base? Eating behavior is an important part of an ecosystem and its proper functioning. There are many factors that affect the eating behaviors in an ecosystem. There are some abiotic factors, such as the pH of the water, temperature, and sunlight availability. There's also biotic factors, such as predator-prey relationships. In our experiment, we wanted to test the pH effects on the eating behavior of dragonfly larvae. pH is a measurement of the alkalinity and acidity of water. It's measured on a scale of 0 to 14. The reason we're interested in the effects of pH on eating behaviors is because if there's an alteration in the eating behavior, this could potentially disrupt the energy flow and chemical cycling in our given aquatic ecosystem. Recently, there's been an increase in runoff from chemical plants, mining, urbanization, and industry that affects the pH of the water in the streams.
For our experiment, we'll be testing the effect of 3 different simulated aquatic environments with varying pH levels on the eating behaviors of dragonfly larvae. We will use dragonfly larvae because they are non-microscopic aquatic organisms that exhibit observable eating behaviors. And more importantly, they're very vulnerable to slight changes in the environment, which makes them great tests. We'll be using brine shrimp as the food source for the dragonfly larvae because they're naturally part of their diet.
First, we will add water to 3 separate containers, making 3 distinct aquatic environments. We will then add 10 drops of hydrochloric acid, HCl, to the first container, bringing it into the pH range of 6 to 7. This will serve as our acidic environment. Then we will add 10 drops of potassium hydroxide, KOH, to the second container in order to bring it to the pH range of 8 to 9. This will serve as our basic environment. We will leave the final container alone and it will serve as our neutral environment. We will add the dragonfly larvae and allow them to sit for 5 minutes to adjust to their new environments. Then we will add 5 brine shrimp to each container and allow consumption to take place for 10 minutes. We will then repeat this process a total of 3 times for a total of 35 minutes. Over the 35-minute interval, we will gather the quantitative data in regards to consumption levels. This concludes our experiment.
Since dragonfly larvae typically thrive around a neutral pH, we expect consumption levels to be highest under these conditions. This experiment has biological relevance because if the results deviate significantly from our expected results, this could have a major impact on our current understanding of the ecosystems inhabited by dragonfly larvae. This experiment could also potentially inspire many new studies, especially if our end results deviate from our expected results. This would mean that there must be certain benefits that the larvae's current habitats have that outweigh the benefit of higher consumption levels in the environment with an altered pH. New studies could be done to explore these opportunity costs.
We believe the results of this experiment will have significant impacts on environmental advancement. Therefore, as a group, we'd appreciate your